Yes, I welcome all of you to this post-lunch session of this international webinar. We have today Dr. Anil K Suresh with us. Dr. Anil K Suresh is an associate professor in the Department of Biological Sciences, School of Engineering and Applied Sciences, SRM University, Andhra Pradesh. He obtained his PhD in biotechnology in 2007 from CSIR NCL, affiliated to Pune University. He was a postdoctoral fellow at McGill University, Canada, and a research associate at the Oak Ridge National Laboratory, USA. He worked as a staff scientist at City of Hope, California, USA. His research focuses on biomedical and sustainable applications of engineering nanoparticles. Dr. Anil has authored over 46 high-impact factor international journal publications, and has 5 patents, 2 books, 18 book chapters. He's a recipient of many awards. Dr. Anil, do you want to correct us?
No. Yeah, no, it's fine. You can skip. Let's start the talk.
He's a recipient of just one more sentence. He's a recipient of many awards, including gold medal for outstanding scientist in nano science and nanotechnology from Andhra Pradesh Association of Biotechnology and Pharmacy. Dr. Anil also is an editorial board member for many international journals. With those words, I request Dr. Anil to talk on sustainable biotechnology, where bio meets nano in harmony with nature. Over to you, Dr. Anil.
Thank you, Dr. Priya. Thank you so much. Those many words were not necessary, but let's see how we can share the knowledge that we have been generating through science. So, am I? Hello, can you see my slides?
Yes.
Okay. Am I audible?
Yes.
Oh, and now you can see my slides also, right?
Yes.
Okay, super. So I'm sorry for this small technical delay. It asked me to go to settings, and then it asked me to add this. Okay, so a very good afternoon to all of you. Okay, so today I'll be talking to you about how my journey started doing nanotechnology to now doing sustainable nanotechnology. So I began my journey into nanoscience and nanotechnology with my PhD. So during my PhD, I learned to make nanomaterials. So I learned to make nanomaterials using healer ways. So earlier people were trying to use earlier people were trying to use chemical ways of producing or physical ways of producing these nanomaterials. But during my PhD, we just transformed a bit ahead, and we thought, why not make use of microorganisms, and that's why I started making nanomaterials using microorganisms. And then after my
could you make it full screen?
Yeah. Can you see now?
Yes.
Okay, so after my PhD, then I started to learn these nanomaterials much further. So in terms of their interaction with microorganisms, in terms of their interaction with cells, in terms of their interaction with plants, tissues, in terms of how these nanomaterials can be used as a catalytic moiety to target drugs, or how these nanoparticles can be used for imaging and so on. So 10 years I have spent learning about these nanomaterials and using these nanomaterials in various drug delivery, it may be, or imaging, or cancer diagnosis, or selective targeting, and so on and so on. So in 2018, all of a sudden, FDA said that the use of nanoparticles, especially for clinical purposes, should be minimized, and there were so many drugs that were banned or cancelled by FDA, and all these gold nanoformulated drugs, or all these iron oxide formatted drugs. Most of the drugs that people have been working on for the last few years, they were all at hold, and a big question mark came on whether we should go ahead and use these nanomaterials for biomedicine or not. So we reached to that state of point. Even now, it's very strict and very stringent, and on what kind of particles that we should be picking for clinical purposes. So in that process, I thought I was thinking on how to convert my knowledge of nano and simultaneously not do something with critical research? And what should I do? So in that process, in that process of evolution, I learned something about supported catalyst. So I think I'll skip the introduction here. Okay, I'll come back directly to the topic or to the story that I want to tell you today. I'll tell you two stories. The first story that I'll tell you about is a supported catalyst story. So this supported catalyst is nothing but is just a support, any neutral support or inert support onto which your nanoparticles are now drafted. So now you should ask me why these nanoparticles are drafted onto your support. See, nanoparticles are drafted onto your support because after any given chemical reaction, may it be a pharmaceutical relevant reaction or may it be an industry relevant environmental applicable reaction, when a nanoparticles act as a catalyst and when the reaction is done, we should have a way to take out these nanoparticles from the reaction. So because neither the pharmaceutical molecules nor the environmental samples uh, can retain these nanoparticles. They need to be purified from the product. That is one very reason why these nanocatalysts should be extracted after the job is done. So in that process, supported catalyst has been evolving, where these nanoparticles are grafted or made on a inert support, which can allow us to hold these nanoparticles intact and allow us to remove these nanoparticles from the reaction post catalysis. So now that is where my journey into supported catalyst, and that is where my journey into how we can make use of support to graft these nanomaterials started. And then I bought something called as chicken egg waste into my laboratory. So as we all know that chicken egg is drastically consumed in so many food stalls and so many restaurants, and after that these eggshells are usually thrown into the environment. And these eggshells, because they also contain food content, because they also contain rich minerals and food content, so it is always prone to infections and stuff. So otherwise they also cause foul smell and land occupants and so many things. But anyway, that is not the point here. The point here is that this eggshell, on the surface of this eggshell, of course we all know that eggshell is made up of calcium, and then on the surface of this eggshell we have these protein layer. We have this protein layer, and I know that microorganisms can make nanomaterials, and I also know that proteins and biological molecules can also make nanomaterials. So knowing that, I thought, why not we make use of this intact egg shell, which is a waste, and see if this can make a supported catalyst for us. So now when I treated as you can see here, when I treated my pristine eggshell waste with aurichloride, which is a precursor for making gold nanomaterials, within 6 hours at 37 degree centigrade, my prediction was right, and these proteins on the surface of these nanomaterials act as a reducing agent, and successfully could make the biggest catalyst in the world, which we named mega catalyst. Now you should ask me, why did I make this biggest supported catalyst using eggshell particularly? So now going back to the chemistry of this eggshell is biological membrane. This eggshell is made up of calcium as well, and this eggshell is highly porous. That means it can allow the exchange of reactants in and out of this pores. And for any given catalytic reaction to happen at a faster pace or at a large volume capacity, you should have a constant allowance of these reactants in and out of these catalysts. So such properties of this eggshell allowed me to think of three major reactions that are need of the day. So may it be a gram scale production of pharmaceutical precursors, or may it be degradation of dyes at extremely large volumes. And that's what we did. I mean, first let me show the video of how these particles were beautifully formed onto the surface of this eggshell without doing anything. No chemicals added, no reducing agents, nothing. Just simply eggshell, egg white, and naturally it made us these gold nanoparticles. And then these are the characteristics of the. I mean, after the particles are made on the surface of these eggshell, we should make sure that these particles are formed and these particles are stable, and that these nanoparticles are gold and they are crystalline. So and we should also understand the size and shape distributions of these particles. So there are basic characterizations that we do. First is based on the color, where your egg white becomes deep purple and the color intensity increases. That is the first indication. And of course, we do XRD. As you can see here, the three peaks that you're seeing here at 111, 200, they correspond to only that clearly says the presence of formation of gold on the surface of these eggshells. And of course, UV visible spectroscopy, gold has absorbance at 520 nanometers, and as can be seen here, you are seeing an absorbance. And XPS again to tell that whatever element that is present on these eggshells is metallic gold, and this confirms that gold is formed on the surface of these nanoparticles. And further we did SEM, scanning electron microscope, to see what size and shape distributions of these particles are formed on your X surface, and this bar graph will tell. I mean, this is of course calculated from this SEM image here, and this bar graph tells us that the particles were somewhere between 6 to 14 nanometers. That means they were very small particles that were naturally formed on the surface of this eggshell. So now, as I just told you, why and how this eggshell is making these nanoparticles on the surface is because of these hydroxyl amino acids that are present on the surface of your eggshell, which are, of course, a protein layer is present, and to this protein layer there are so many hydroxyl amino acids, and these hydroxyl amino acids act as protecting agents, and they give away H+, and in that process the gold gets reduced automatically on the surface of your eggshell. Now coming to the key applications of where this eggshell can be utilized. See, the first application that I demonstrated was by collecting sewage textile dyes from the environment. See, as we all know that these dyes that we use to ink our textile garments are extremely carcinogenic and extremely toxic to the environment, not only humans, but also they suffocate aquatic and environmental beings. So now the dye that goes out of these textile dyes should somehow needs to be cleaved or needs to be detoxified, and that can be done by using gold nanocatalyst. And now here the advantage is that the same catalyst can be done now, of course, with the gold, but here now, because this gold is supported onto eggshell, this can be easily removed from the reaction after the catalyst is done. Okay, so now when we did the reaction, as you can see here, before this use of eggshell catalyst, of course, there were so many papers that were published on the use of supported catalyst to demonstrate the degradation of sewage dyes. So the farthest or the highest volume that was degraded was only 100 ml, and by using a catalyst that was generated using hydrogen. So now that catalyst was generated from a plant precursor where the plant was processed and the plant was refined. It was a 24-hour process, after which they made a hydrogel, and onto which they grafted these graphene sheets, and onto which they loaded your platinum nanoparticles, and that was 1 centimeter by 1 centimeter. And using that catalyst, they could degrade 100 ml of dye, 100 ml of dye only at a concentration of 30 milligrams per liter. But to the surprise, our catalyst can degrade 2,800 liters of dye. Only one supported catalyst that we made using one egg could degrade 2,800 liters of a dye at 200 ml cycles. Of course, the dye concentration again same, 30 milligrams per liter. So let me just show you a video of how efficiently the dye degradation was happening for these textile sewage dyes. So as you can see, the dye was coming from here. These are the catalysts, and you can see the detoxified clean water that has been completely detoxified and free of toxic azo dyes. Okay, now this is a comparative. This is a small comparative table to talk about how our catalyst is efficient with respect to the rest of the catalysts that are already there in the literature. As I just told you, our catalyst can do up to 14 cycles of 200 ml. It is hand recyclable, and we could do multiple dyes, all the various forms or various combination of cationic, anionic, and other forms of dyes as well. And second application that we wanted to achieve is gram scale hydrogen of nitrogen. It's a paracetamol. The drug that we use when we get common fever is paracetamol, and that paracetamol is produced by reducing a precursor called as para-nitrophenol. So this para-nitrophenol gets hydrogenated to produce para-aminophenol, and that undergoes the acetylation to give us this paracetamol. But the problem is how do you produce this paracetamol in gram scales? Today we import these precursors from China. So in a way to not import these pharmaceutical precursors from China, and to demonstrate that we can no doubt produce, we can no doubt produce these pharmaceutical precursors at the gram scale. So we use our catalyst, and for the first time in the world we showed that up to 1 gram quantities of pharmaceutically relevant precursors can be produced indigenously, and that too using a catalyst that is being generated from excel waste. And before this, the highest volume, the highest amount of drug that was produced was, of course, in milligrams, 0.091 milligram, 12 milligrams, 1 milligram, and so on. They're all in milligram quantities. But for the first time, we could generate 1 gram of this catalyst, which is a big breakthrough in terms of pharmaceutical sectors for the generation of indigenous precursors. And not only that, because this catalyst was handy, it was big in size, it was handy, right? So therefore, after the completion of all the degradations, after the completion of all the generation of pharmaceutically relevant precursor molecules, the catalyst or the gold, because it is expensive, it could be simply retrieved back from this support by dipping it in aqua regia. So we started from egg shell, and we came back to eggshell. So that's how the story of eggshell started, and the story of eggshell ended. But in that process, this eggshell could do large volume degradations, could do gram scale production of pharmaceutical precursors. So and if you see the advantages of such a work, so we use bio waste as a support, and the synthesis is autogenic. We are not using any reducing agents or any other factors to make these particles. The synthesis is ecologically friendly, very easy to make, and large support area. We can use it for fixed bed catalysis. It is easily removable from the reaction. Highly stable. Years of sulfur. These are all the important characteristic properties that are pertaining to our catalyst. And we have patented this. Work as an Indian patent, and this work was recently published in the Journal of Applied Materials and Interface. And keeping in mind the impact of or novelty of this work, the world's renowned magazine called as Chemical and Engineers also highlighted our work in their magazine as a news article. And I'm also funded with DBT, which is called as Accelerated Translational Research Grant. So when you have something which is innovative, and DBT usually funds for such innovative work, so that they can be commercialized and bought into industrial practice. And by utilizing this fund, I will be demonstrating the same reaction at extremely large scale, pilot scale, up to capacities of 500 liters. And this is a setup that we have established for that. And I just got this funding, and I'm thinking about next 6 to 7 months, where I'll be demonstrating the degradation as well as pharmaceutical production of pharmaceutically relevant precursor at large volumes, and will open up my own company and name of Sustainable Science Solutions. And I'm trying to bring this to people through my company itself. I don't want to give it to somebody else. Okay, so now coming back to the second story that I want to tell you today is my recent development of a biofilm called as, we named it as spec film. Okay, I'll tell you what it is. So again, see, when I was working on this eggshell, I thought, okay, why not bring in other waste and see if we can make another form of supported catalyst. So in that process, I bought fish scales, because I know that fish scales also contain collagen and proteinaceous content in them, and they can also be very good supports for large scale catalysis. So with that intention, I bought fish scale. But when I bought these fish scales into the lab, and when my students were cleaning these fish scales, they learned that the fish scale is becoming transparent as we keep on cleaning them. So as you can see here, this image showing demonstrating the transparency of our scales. So now when the fish scales were becoming transparent, I told my students to just take the fish scale and why don't you put it in the spectrometer and see if you can and do an analysis for any given analytes to replace plastic weights. So then in that process, the birth of this spec film happened. It's a beautiful story. And you all know, I'm sure you all know what is the use spectrophotometer. It's a machine that can analyze any given analytes or any given chromophores by absorbing a particular wavelength of light, right? And spectrophotometer basically needs a cell called as a cuvette to load your samples, and so that the measurements can be taken. And that cuvette is usually made up of plastic. So, and we don't want plastic because we are already contaminating our Mother Earth or Earth with so much of plastic. And as scientists, we're also liberating or using so much of plastic, and we want to avoid plastic somehow. And therefore we thought, can this transparency of this fish scales can be used to replace cuvettes and do spectroscopy analysis of various analytes. And how did it begin? Because I am saying, right, it's a film. So now when something is a film, how will you insert that into spectrophotometer? So you definitely need an adapter for that. So we custom designed this adapter by ourselves in our laboratory, and we 3D printed this using a 3D printing machine in-house at our facility. And as you can see here, this is how your film can be inserted into your adapter. And after it is inserted into adapter, we just take one drop of this analyte and place it in the position where the light beam from the spectrometer passes through that. So there is a particular position in the spectrophotometer where the light is passed. So that spot we identified as given here with these dimensions. This we got it from the manual, and accordingly we have spotted this, and we have used that spot to analyze our drop test our analyte. So and why, and you can ask me why your fish scales are retaining this drop onto this analyte for such a long duration. So this video I'll just play for you, which you can see it's a 45-minute video, but for that duration the drop was staying on intact onto this biofilm. So this is the drop that we casted onto the film, and as you can see here, the drop stays there and it doesn't drip from there, which giving us enough time or enough room to take this back and put it into the spectrometer and do our spectroscopy analysis. And the reason for the stability of this drop on the film is because of the structure. I'll show you. I think I skipped that slide. Actually, these scales have so much. As you can see, these scales have so many grooves on their surface. I think I missed my SEM slide. So they have grooves on their surface, and those grooves actually allowed the holding of these samples onto them very firmly. Okay, so now I mean, as you all know, for any given analyte to be able to measure using a spectrophotometer, it has to be completely transparent. So now, and we assess the transparency of this film in the for a spectroscopy analysis, and we compare that with quartz cuvette, glass cuvette, plastic cuvettes, microplate, as well as our template. So as you can see here, the transparency for our biotemplate was matching by par with that of plastic cuvette within the window of 360 to 900 nanometer. That means any given analyte within this window can now be analyzed using our biotemplate. And the transparency was up to 85%. So that was also matching exactly to that of your plastic cuvette, which gave us a sign that now these can be used to replace plastic and can be used for analyzing various biological analytes. So we started with nanoparticles, we started with BSA, we started with metal chromophores, we started with various colored compounds that we have in our laboratory, and we demonstrated that. And we also demonstrated the thresholds of the sensitivity of these analysis. What the least volume that I can use to measure using my spectrum. What is the highest volume that I can use, after which it dips off. So the least volume that we can go was up to 0.25 microliter, which is not possible at all using your cuvette, because cuvette needs at least 500 microliter, or if it is a small cuvette, then at least 100 microliter. So 0.25 microliter analysis is very, very essential, especially when you are dealing with precious analytes, narcotics it may be, or explosives or TNT. When you deal with such precious analytes, low volume spectroscopy becomes extremely ideal. So now using our spectrum, one can now go and measure the least volumes. And we also determined the concentration threshold by using one of the dyes, and the least that we could go was 10 micrometers. Of course, this is for this particular variant, but that can be designed, and that can be standards for any given analyte, and can be standardized. And likewise, we also demonstrated on template protein staining, which instead of fluid now, or instead of 97 plate, we just we added our protein onto the surface of our film. We took a dye and we added this onto the surface of our film, and we could match the analysis in par with that of your 96 well plate. And we also demonstrate the large scale production of these fish scale templates. So how the templates are derived, initially your fish scales are like this, and how your transparent films are derived is just by simple etching. So we just do stepwise NaOH treatments as well as HCl treatments, which carefully removes the minerals and calcium layer or other deposits from the surface, and this is calcium, which is the skeleton of your scale, which gives so much transparency to your fish scales, which is over 80% and within the window of 400 to 900 nanometers. And the best part is, after you complete the analysis, spectroscope analysis, because this is derived from nature, this is a food waste, so this possesses the ability to self-degrade within 42 days. So that means within 42 days, this film can completely be degraded into or vanishes. And that's how that's how this slide shows the demonstration of such a vanishing of your biodegradable spec film. Again, if you ask me what are the uniquenesses that goes with this story is, we bought waste, and from waste we could generate a transparent film that can allow us to do low volume spectroscopy of less than 10 microliters, and the least that we could do is 0.25 microliters. And and finally the template is biodegradable and can be thrown back into the environment without causing much of a pollution, unlike these plastic. So now we are in the process of converting this film into giving it a shape of a cuvette. So we are performing these experiments in the lab, where we are trying to cast or mold these cuvette shapes, or give this film a 90-12 plate or cuvette shape, so that eventually we can replace these plastic cuvettes and 96 plates. So this work is also patented recently, and the work is published in the prestigious Journal of Green Chemistry. And to my fortune, this work also is recognized by CNN, and this work is highlighted by them as a news magazine in CNN as well. So this in my talk, and that's how I evolved my nano research into doing sustainable nano. That's why my title goes: Sustainable Nano, do research that is safe to the environment and also safe to the humans. Thank you so much.
Thank you, Dr. Anil. It was a wonderful talk. The talk is now open for discussion. If there are any questions, please do come forward. Dr. Anil, it's a wonderful talk, and it's all like translational, lot of translational potential with your study. But I just want you to just give a glimpse of what happens with the cost. You have the nanoparticle conjugated eggshell, as well as this fish,
Yes.
Nano microplates and things like that for application process. When you compare with the standard protocols for such applications, what cost benefit you will be having?
Okay, that's actually a very good question. That is the reviewer asked me that question actually. What is the cost comparison for your technology with what we have in the market? So actually, to be able to generate that template that I was showing you, it costed me only 0.2 paisa. So, and whereas your plastic cuvettes cost 4,000 rupees. So each cuvette will cost you 4,000 in a box. You get 50 cuvettes. So 4,000 divided by 50 is the cost for that cuvette. But whereas the spectrum that I generated costs only 0.2 paisa, very affordable. And most of it is coming from waste. So by doing so, we are generating revenue out of waste. So that means we are creating a demand for a waste. Otherwise this waste is just dumped into the environment, and the aftermath has to be seen: pollution, smell diseases, spread of diseases, and so many things are that they are following from these waste. So it's not about just the price, but we're also creating a demand for these waste now. So by bringing these waste into the translational research.
Yeah, that's interesting. Anyway, that's a very good presentation with a lot of thoughts for the students also. Like, there are many students, I'm sure, are working with nanoparticles, and this is one of the best examples for this translational potential. So thank you,
Yeah,
Doctor.
actually, I have PhD positions in my lab. If somebody is interested, then they can definitely write to me.
Yeah, that's great. So
yeah,
it's an open call. Now, thank you, Dr. Anil. Shall we move on to the next speaker?
Yeah. Thank you so much.